My name is John Hagen. I'm an independent fish biologist.
I'm Bryce O'Conor and I'm a fisheries biologist at Trout Environmental.
This study is primarily a study of the genetic diversity of bulltrout in the Williston Reservoir Watershed. To understand population structure requires genetic studies. What we're here doing is part of a larger genetic study of the distribution of genetic diversity around the Williston Reservoir Watershed for bulltrout. The second objective is a study of age, life history, and growth. Bulltrout on their spawning migration tend to gather together in quite vulnerable aggregations. They're not spread evenly, typically, across the entire length of stream. The Missinchinka has over 100 kilometers of accessible stream length that bulltrout can use. And the population probably is comprised of less than a couple hundred individuals, perhaps much less than that on some years. So that population of adults spread over that distance, we wouldn't be able to find them efficiently by just hiking along and fishing. So we use the helicopter for that, and we spot groups of fish, or at least a few of them from the air. They're difficult to spot, especially in the Missinchinka. And when we find them, we set down and fish for them. The steps we need to take when we catch a fish are we anesthetize the fish for about 5 minutes in a solution of clove oil and water. First step is to take a couple of plugs, tissue plugs, from the fish's back. Those are removed and then they'll be frozen tonight, and we'll keep them frozen until they make it to the laboratory for analysis for mercury and other metals. That has been a byproduct of reservoir formation. It's getting a little bit better, but that's something that's of very high interest to the nations because these are an important food fish. The next step, the most important step in this particular case, is to take a very small clip of the pelvic or adipose fin and preserve that in alcohol. That's the genetic sample. So that will be sent to the laboratory at UNBC, and once the samples are compiled and a student is arranged, then the lab analysis will start. Third, we cut into the pelvic fin ray. We'll take the first two pelvic fin rays and only a section, not rip off the whole fin ray, but just take a section, because that section that we cut out will heal over. And that section of fin ray will be dried and then sent to also to a lab for analysis for aging. Finally, and importantly, we'll measure the length of the fish and weigh it. The heaviness of the fish for its length is its condition factor. So that is a basis that we can compare to the past to evaluate how the fish are doing in terms of growth. Once the fish has been processed, it's back into a recovery tube, rubberized fabric with flow-through ends. It's anchored in the stream, so there's oxygenated water flowing through the tube. The fish is allowed to recover for 15 minutes, and then by then it's usually regained full vigor, and then we'll release it back into the pool. The rationale for these projects is that the spatial structure, the population spatial structure for bulltrout, is not understood with any real data. There's also the question of the deeper ancestry of Williston bulltrout. Where did they come from? The recent and ancient history of these fish, they can't tell us. The genetic information can.
It's kind of like a critical part of the whole picture, how these populations are related to each other and how they interact to create the metapopulation of the whole Williston Reservoir or Upper Peace River. It's kind of a cool situation because Tsay Keh has, over time, taken over the ongoing monitoring, conservation, and and stewardship of bulltrout in the Williston Reservoir from BC government and in partnership with the BC government and other First Nations. So it's great that Tsay Keh is leading it, also with support and partnership from other nations, because it just gives that base information that allows us to make the right decisions when it comes to bulltrout conservation.